Hello and welcome everybody to our webinar in Vitro tools for predicting biopharmaceutical performance on drugs in vivo. My name is Ali and I will be your host for the webinar today and all the following webinars within this webinar series on permeability. On today's agenda, we have many interesting topics, beginning with the basic question on why to use in vitro tools to predict drugs in vivo performances. Then we are going to continue with the explanation of an important indicator, the apparent permeability or Papp. And we will conclude the presentation with a lot of informative and application close example. We are very happy to have Professor Massimiliano Pio di Cagno as our presenter for today's live webinar. Max has a very impressive academic vita and can therefore be called a total expert in pharmaceutics and especially in drug formulation and permeability. Next to that, Max is also a freelancing scientific consultant for Inome in Germany, the company which manufactures and commercializes PermeaPad. Max co-invented this product together with Professor Annette Bauer-Brandelbeck at the Southern Denmark University, where he used to work. As you can see, I did not overstate by calling Max a total expert. So before we start with the webinar, a few little things regarding the organization. We have opened a Q&A window where you can type in your questions. And we have also a chat. If you have any problems with technical issues or sounds or visuals, you can type something in, or you can reconnect the webinar and you will come back to it immediately. This solves most of the problems. And at the end of the presentation, I will come back and we will answer all your questions from the Q&A together with Max. And without further ado, I give the word to Max. Enjoy the presentation and see you at the end again.
Yes. Thank you, Ali, for the nice introduction. And I hope that all of you can hear me well. And thanks to all the attendees of this of the webinar of today. So as Ali has stated already, the goal today is to present to you the commercially available in vitro tools for predicting biopharmaceutical performance of drugs in vivo. So I will start with try to contextualize why this is actually important for pharmaceutical purposes, and I will try to contextualize in vitro in tools in socioeconomic perspective. I will repeat some of the concepts discussed during the first webinar of the series related to the apparent permeability coefficient and its meaning. I will then focus on the commercially available artificial in vitro barrier and setups that are possible to utilize, and I will then conclude with some examples on applications for both enabling formulation, but also just in vivo prediction of permeability and in vivo performance of active ingredients or new chemical entities. So starting with the general contextualization, put the in vitro testing in the context. So why we believe that a series on permeability is needed and was needed? Well, as most of you probably are aware, when it comes to prediction or in vivo performance of drugs, still pharmaceutics and the pharmaceutical industry is still heavily using in vivo studies. Especially when it comes to formulation and now enabling formulation, the only way to predict the reliable data and generate in vivo data is to utilize animal. So, but is there a different way? Is it possible to do something to make maybe the animal a little bit happier than the situation and the current situation? That's why we in Fabioc believe that the permeability concept is still not implemented enough, and probably because also sometimes is misinterpreted, and this has been widely discussed in the previous webinar. But definitely another problem is that in vitro methods are not yet implemented as they probably should, and also at the light that in vitro tools such as PermeaPad, but maybe others, can now provide and generate data that somehow can be predictive for in vivo situations. And it's not just a ethical issue. So reduction of animal experiment that definitely is and should be also our goal. We have also an economical a huge benefit by the possible implementation of these in vitro studies. And if we consider a very general timeline of modern drug development, in average, the development of a drug from discovery up to formulation can take up to 10 to 15 years in average. And this developing process is clearly divided in two phases: a preclinical stage, where actually the drug and the formulation are developed, and then there is a clinical stage. And surprisingly enough, both costs for preclinical and clinical has been growing exponentially from the early 70s. This is a picture that stopped in 2010, but this line has been growing and growing. And what in a way it's understandable is that the clinical costs are very high, but the only way to justify this very high cost in preclinical testing is animal testing. Animal testing are expensive, and most of them are, there is a generally overuse of animal testing. And just to give some numbers, just in the US in 2020, more than 100 million animals has been sacrificed for the purpose of pharmaceutical development. And this is the same picture in Europe and maybe other countries in all over the world. So how do we see the role of in vitro testing in the nearest future? Well, definitely reducing the cost and time of preclinical drug development. Very important, increasing of understanding. We used to say, put science into pharmaceutical science. So try to have a better mechanistic comprehension of the process of drug absorption and how the formulations we produce work. Providing useful parameter for in silico drug profiling, because in silico drug profiling without proper parameters cannot really work. And I mean, it's not just us, but it's legislators that want us to go in vitro to refine, replace, and reduce animal testing. This is in the law of Europe. Europe and FDA, they put the target 2025 is not going to be probably possible, but already from 2013, we have a forbidding in testing cosmetics to animal. So it's not a matter of if, it's just a matter of when, and we need to be prepared as pharmaceutical world on a wall. So definitely we cannot rely anymore on nocturnal water partition coefficient, but luckily in the last years, a very cool concept has been introduced, the concept of permeability. And I will just now very briefly go through concept that has been already discussed in the previous webinar. So the concept that in permeability testing, the basic principle of permeability testing is to measure the apparent permeability coefficient. All the permeability testings and setups have a general structure. There is a donor chamber, there is a barrier, and then there is an acceptor chamber that can be or not steered accordingly to the setup we have. The drug or the formulation is placed in the donor compartment, and then the drug molecules will start to diffuse spontaneously through a chemical gradient process, activity gradient process, so spontaneous process. And this can be approximated by utilizing the Fick's law of diffusion, even though we have a transition of different phases, and by accounting for the flux, so the amount of drug that accumulates over time in the acceptor, we can then measure the coefficient called apparent permeability coefficient. The major difference between the flux we measure and the permeability is that we normalize the flux over the initial concentration of the drug in order to have a parameter that is independent from the concentration and can therefore be compared. The goal for this kind of in vitro assays is, of course, to get somehow what it has been introduced by Amidon when and coworkers when they have introduced the BCS classification system. So have a parameter, upper impermeability, that can correlate to the fraction absorbed with this kind of profile. Or in other words, if we plot the logarithm of the fraction absorbed over the permeability, we should obtain a sort of a steady line or a straight line. And we don't want to achieve just for the drug and the active ingredients. What we are most interesting is, can we use enabling formulation? Can we get information on our formulation is if we are actually doing something that is positive for in terms of in vivo performance later on. So coming back to the going forward to the experimental setups, we always have a donor compartment, a barrier, and a septo compartment, and we can actually utilize different type of setups. Vertical diffusion cells. This is the picture of front cell for Permegear, for example, where one type of biomimetic barrier, in this case PermeaPad, was placed in the between of the donor and the acceptor compartment. We can use side-by-side diffusion cells, or if we are interested in high throughput screening, then definitely multi-well plates, 96 or 24, but the concept is always the same. We have a donor compartment, an acceptor compartment, and a biomimetic barrier in between. So, which is the picture? Well, the correct picture of implemented methods is that we have cellular barriers and artificial barriers that are available. The cellular barriers are actually have an advantage, and that comes from the possibility of accounting for the care-remitted transport. So if we have a drug that has a significant active transport or facilitated transport through aquaporin, for example, then cellular barrier assays will actually account for it, whereas artificial barrier focus mostly on passive transmembrane and partially also on paracellular pathways of drug absorption. Definitely the most used and implemented in vitro cellular approach for measuring the permeability is the CARCO2 model. Here I have just here you can find a sort of a picture representing the system. CARCO2 cells, CARCO2 means or stands for heterogeneous human epithelial colorectal adenocarcinoma cells. Basically, these are cells that have been extracted by a patient suffering from adenocarcinoma, and that's been and that can be harvested on demand, so when it's needed. And these cells has a peculiarity. They grow with a basolateral side and an apical side, exactly as it would be in the intestine of a healthy person, growing on a monolayer. And this is actually very good for permeation testing because, of course, we can account for the flux or the so-called influx, but also, if significant, we can also account for the efflux. If, for example, there are expression of P-glycoproteins that will take the drug from the basolateral side and bring it back to the apical side. So what we measure is basically the net permeability, but there are possibility to block on both sides the transporters with the targeted inhibitors to quantify how much is influenced in the net permeability from the active transport. So this model has huge limitations when it comes to high throughput screening, or even more when it comes to formulation testing. First of all, we have long harvesting times and we need a dedicated lab. There are reports of shorter time, up to 3 days for harvesting cells for this kind of studies, but what it has been reported is also that in this kind of fast growing cells, the expression of the proteins is much reduced. There is high variability, so data cannot be really compared between different labs, and poor reproducibility. But these models definitely account for active transport. So if you have a drug that has a significant active transport, then that can be useful. We should remind that the passive transport is in most of the case, the most significant, because it's just thermodynamics. It will always take place for any drugs. And then we have the artificial barriers. And here, definitely, we should start from the most implemented one, the PAMPA, so the Parallel Artificial Membranes Permeability Assay. This has been developed back in 1998. It was an industrial development process. And that's actually this is actually reflected in its high throughput screening nature. So this model has been developed for testing the most possible compounds in high throughput screening setup. This barrier is actually a very simple and elegant model. It's a filter that is soaked with organic solvent in which some lipids are dissolved. The original composition was endodecaying solution with egg lecithin and other lipids such as phosphatidylcholine, phosphatidylethanolamine, phosphatidylinositol, and cholesterol. And generally, in this kind of setup that is on, as I said, 96 well plates setup, the donor chamber is the chamber in the bottom, and sampling in the acceptor are made on the top, and this also is made to facilitate the sampling. To a certain extent, some of these PAMPA barrier can be used for enabling formulation testing because they can sustain some excipient and some solvents. But this model definitely has difficulties in account, for example, of paracellular transport because it is a partition-driven process. So the drug will have to partitioning into the barrier, so a lipophilic environment, and then again partitioning again in the acceptor. And this process can take time accordingly to the drug. And here I summarize, or I actually borrow this table from a very nice review on in vitro tools for permeability testing published in 2018 from Berben and co-workers. And here are listed the different type of the PAMPA, so from its original product till the evolution of more mimicking specific tissues like BBB, the PAMPA skin, and here also the different composition. All these models, since it's a partitioning, require some incubation time that can also be pretty long, and most of these barriers also need an impregnation time that can go, that require 60 minutes, for example, of soaking the filter with the solution before you can actually utilize it. Not all, of course, of these barriers. Some are ready to use. And when it comes to the ability of this barrier to sustain enabling formulation, there are some interesting reports in the literature. I guess that it's not so easy to find information because PAMPA is highly implemented at industrial level. So it's maybe the publication rate is also a bit lower than other models, for example, the PVPA. But this nice study show, for example, the case of etoposine. Etoposine is a drug that is a BCS class 4, so poorly soluble and poorly permeable. And in these studies from Bang et al., they tried to solubilize the drug utilizing tocopherol pegylated, TPGS. And as you can see here, above the CMC of TPGS, 0.3 milligram per milliliter, you have a significant enhancement of the apparent solubility of the drug. Testing the formulations on the PAMPA resulted in a very strong decreasing, actually, of the permeability when the formulation was applied. And this is actually understandable because the solubilization is done by micellization. So the etoposine molecules will be incorporated in the micelles, and they will not be not available directly for creating the gradient of concentration that push the drug through the barrier. And this data actually, interestingly, reassembled partially what has been then shown in vivo. So to some extent, with these specific excipients, PAMPA somehow proved to be able to somehow predict the role of a formulation in vivo situation, even though this is not really bioavailability data, but this is permeability through the in vivo rat tissues. And then we come to another barrier that is definitely the barrier where I'm most familiar with, and this is or another type of assay that is called PermeaPad. This is kind of a schematic representation of what PermeaPad is. PermeaPad is the barrier is composed by two low retention layers of regenerated cellulose that comprise a phospholipid layer of phosphatidylcholine S100. This is commercially available in two formats, in pads that can be used for any type of diffusion cells, but also now, since 1 year now, in high throughput screening, so in 96 well plates format. The big changing or the big advantage of this barrier is that due to its design, it owns extremely high chemical and mechanical resistance. And also it can account also, or we believe it does, for also the paracellular transport. So if we look at the formats, we have pads. This is how the pads looks like when you actually buy it. So they are dry, and this is how the multi-well plates is structured. So if we have a bottom plate that is actually the acceptor room in general, then we have the insert plates where the barrier is glued, and then we have a lid to cover. And what is happening when we take the barrier? So the barrier is also very good because it's ready to use. So in the moment you place the barrier inside, in between your receptor and donor chamber in a vertical or in a side-by-side diffusion cells, you have an instantaneously formation of what it has been defined as a liposomal gel. So basically, liposomes are spontaneously forming, and then they press each other, once to each other. So this generates an interesting phenomena that we have some interstices in between where small hydrophilic molecules could permeate, as well as in real life when it comes to paracellular transport. And if we, for example, compare some drugs that are sensitive to paracellular transport with PAMPA and PermeaPad, we can see that PermeaPad in this case is a bit more, it's a bit better in predicting. So this is a picture that is taken from bioassays. There are, since it has been, it's rather implemented method. There are several companies that provide services in measuring permeability utilizing the pump. And this figure shows the measurement of permeability of 6 different drugs in which we contain caffeine. So if we look at this one, we would definitely believe that permeability of caffeine is not that good. So we would anticipate a poor permeability. But caffeine has 100% bioavailability. And this is actually what we measure with the PermeaPad. This was experiment made with the pads, but as you see, very permeable, very bioavailable compounds shows a very high permeability, while very poor permeable compounds like atenolol, or even though calcine is a compound that is not really a drug, but is a very poorly permeable compound, shows to be very, very low permeable. So in terms of biomimesis, PermeaPad might have some advantage in some specific case compared to PAMPA. When it comes to the plates, we also have some interesting feature I would like to discuss with you. So as I said, this is the structure of the plates, and also the plates are a bit different from the one you would consider normally. So this is a drawing taken for a master thesis of a student in Denmark, where actually that reassemble how the shape of the acceptor chambers is. You see, they are not rounded. They have a very peculiar shape. And this is by design, because this allowed the top insert to have space for sampling. So here the syringe can go in and sample from the acceptor compartment, avoiding, for example, the needs of lifting the donor compartment on the top. And then there is another interesting technical features. This is a side picture of the wells, and you see that the barrier is not flat, but it has an angle. And this is also by design, because this angle allowed the steering bar to be placed inside. So we can have a 96 well plates with steering. Here, just a very brief description of the volume. That so we generally we have a maximum volume of 400 microliter in the bottom well, 200 in the upper well, and the sample volume maximum should be around 120. And the barrier is is not polarized, so we can perform experiment from top to bottom and bottom to top. And if some of you has attended the first meeting of the first webinar of the series, I stressed a lot the attention on the importance of the steering. And that's why we believe that it's very important, especially for poorly permeable drug, to have a good steering in the acceptor. And here I show you what I meant, because and this data from student, my former master student Elisabetta, and as you can see, the permeability measured for medium permeable drugs, hydrocortisone, here is compared in different situations of steering. So if we have orbital shaker, or if we have magnetic steering inside the vials, and I mean, this is not, it is a medium permeable drug, and you see how significant can be the impact in reducing the austere water layer. And this is and this is a very good plus in this kind of system, because you can monitor and control it. And coming back a little bit on the comparison between in vitro and in vivo correlation. So here is a nice study from Jacobsen et al. From 2020, so pretty recent. And in these studies, 14 compounds have been tested for their permeability using almost neutral solution of these drugs, and using the 96 well plates PermeaPad. And the data has been then compared with available data for CACO2 and human fraction absorbed. And as you can see, the PermeaPad behave, or the PermeaPad permeability is measured, reassemble very well the CACO2, maybe not in precise absolute values, but this would be surprising if it would. But what is very important is the trend. And as you can see, the comparison within CACO2 and human fraction absorbed is basically the same within PermeaPad and human fraction absorbed. So this is very good. And in a way, PermeaPad is getting close to reassembling the concept described by Amidon back in 1995. So PermeaPad definitely is actually able to predict eventual permeability or bioavailability of drugs, of most of the drugs. And then I would like to stress just one last, I would like to have just a couple of slides on another type of barrier that is reported in the literature that has been utilized for, let's call it permeability studies or diffusion studies. So I think it's more correct, that is the regenerated cellulose barrier. So here, this slide is similar to the one I show in the previous webinar, but I think it's good to show it again, because when it comes to biomimetic properties, we needed to have a correlation between not only the size of the molecules, that obviously takes a big role here, but also partitioning, especially if we have a strong intracellular permeability of the drug. And here I reported the data I have been collecting using regenerated cellulose and different drugs. And here, this is data from another work where they used actually a much smaller cutoff of regenerated cellulose, so 2 kilodalton. But in both cases, you see each one of these blue dots represent drugs, and the permeability in this kind of study is actually nicely related to the size. So in a way, the permeability is proportional of 1 over molecular weight. And this is not reassembling a permeability process, but this is actually reassembling a basic diffusion, where the Brownian motion will produce a diffusivity of the drugs that is inversely proportional to the radius. That's why I'm saying that the cellulose hydrate, or the regenerate cellulose, is not a biomimetic barrier as such. And this is even more crucial when you have a static diffusion. And to stress even more this aspect, I think I can give you some example from my own research. And we have performed some studies on PermeaPad and enabling formulation, very simple enabling formulation, liposomal dispersion of phosphatidylcholine S100 with two different types of drug, caffeine and hydrocortisone. They were chosen because of their different molecular space and properties. Caffeine is a very hydrophilic molecule, it's a hydrophilic molecule with a good solubility in water, which express also very high permeability, while hydrocortisone is a very lipophilic molecule that express poor solubility in water. And we tested the formulation using regenerated cellulose, and actually the results were extremely good because the comparison between the cellulose formulation, so the formulation seems to provide a much higher permeation of the caffeine through the barrier, and even more for the hydrocortisone. But then we tested this with a biomimetic barrier, where the kinetics of permeation is much lower and much more comparable to real condition. And what we got was actually pretty disappointing, because basically with the caffeine, the formulation did not have any positive effect at all. And with hydrocortisone, if there was a positive effect, that was really, really much reduced. So that's why I'm saying that it's important to utilize barrier that are biomimetic properties, because this will also allow you to avoid a false positive, like in this case. I mean, this is not the results we want or we wanted, but that's actually what reassembled then more likely in vivo condition. So in a very brief conclusion, I would say that if we have to compare somehow the properties, definitely the PermeaPad has some advantages compared to PAMPA, some positive aspects, even though PAMPA is still the golden standard when it comes to permeability. And I think that when it comes to CACO2, my general thought, or my general feeling on this is that this could be studied or used if you really have to. So if you know that you are studying a drug that really has significant attention or interest in active transporters. I would just focus on the usage with enabling formulation. There are PermeaPad is definitely the most solid of all, and actually when it comes to PermeaPad, there are some surfactant where, for example, PermeaPad will not sustain chemophore, RH, Solutol, Tritonex, and others, whereas PermeaPad results a little bit higher in terms of mechanical strength. So before concluding, I would just go to the next webinar of the series. We now have two webinars. One, I would say, very theoric, and this one a little bit more divulgative, related to permeability studies. The next one is going to be most focusing on data interpretation in praxis. So what should we do when we collect the data with the data we have, and how to improve our data analysis to get the most information out of the permeability experiment we perform. And with this, I would like to thank you. And yeah, if you have any, if you need any information or anything that is unclear, please contact. Inoa. Yeah.
Thank you, Max. Thank you very much for this presentation. I think we got a pretty clear insight on why to use in vitro tools on predicting in vivo performance. We will now share the link to the slides for all you in the chat. You can download it. We will also send you the recordings afterwards. And we do have some questions that come from the audience. I will read them, and yeah, Max, you can then answer them. The first one is, if permeability of the compound is very low and we can't get a steady state measuring the Papp, can we try to use other parameters to estimate in vivo performance of the formulation?
Yes, thank you for the question. I think that yes, you can. If the permeability is very low, you can just account for the, let's say, fraction permeate, in the sense that you can run an experiment for, I don't know, maybe 12 hours and just get the amount of drug that has permeated over in this period, and use this parameter to compare with, for example, fraction absorbed or your reference experiment, because it's always important to have a reference experiment in this kind of study. For example, a solution of the drug or a suspension of the drug. If we are talking about enabling formulation, of course, yeah.
Mm-hmm. Another question is if there is any impregnation time for PermeaPad.
No, there is not. The barrier kind of it's ready to use. As the moment it's hydrated, then you can start the experiment.
Okay. Another question is, what type of data results do you have to claim that PermeaPad can account the paracellular transport of molecules?
We have, for example, the example of caffeine, and this is the one we believe is for the moment the most relevant, and we are investigating it now. So that's why I'm saying that we believe that it can account for some compounds at least.
Mm-hmm. We have still 2 questions. Can you explain again the relationship between CMC and the reduced permeability through PAMPA?
Yes, when you have an increase, so in the case of micellar formation, lipophilic drugs will be when you go above the CMC, you will start to have micelle formation, and therefore the drug will be incorporated inside the micelles. The micelles are not this incorporation will create a sort of carrying drug system. So the drug molecules will be not available in the water phase directly. So they will not be molecularly dissolved as such, but they will be mostly incorporated in the micelles. And what it has been reported so far is that the gradient of concentration to permeation in general is generated by the molecularly dissolved fraction. So that's why increasing this, in this case, the solubilization induced by the CMC will not bring a positive effect in permeability, but will actually reduce its permeability.
Okay, another question is, are these permeation assays especially good for any BCS class?
Well, I think PAMPA has been proved to be very good in most of the families of the BCS. Probably PermeaPad and both PAMPA will be maybe more effective when it comes to problem in solubilization. So class 2, for example, class 4, but I guess they could probably work with all the systems, yes. Well, what I think it might not work if you have something that interact with the barrier, like if you are interested in skin permeation, for example. Maybe you, in this case, you need to use an ex vivo studies, for example, a tissue-based study.
Okay, another question just popped up. Dominic says, thank you for the nice presentation. Regarding the reproducibility of PermeaPad, how reproducible is the in situ lipoid gale formation, architecture, numbers of vesicles? Can we assume that there will always be comparable extent of transvesicular and paravesicular flux?
Thank you for this. It's a very interesting question. For the data we have, and as said, it's limited because we have just tested a couple of drugs where we know that paracellular transport is very relevant. We see a slightly higher variation when the permeability is very high, but it's pretty reproducible, I would say.
Mm-hmm. Now the last question is from Elisa. She asks if PermeaPad can be used with both small molecules and macromolecules.
For the moment, we have information. The largest molecules, I think we tested is vancomycin. We have measured the small PEG, 400 kilodalton. We know that there is a limitation in size because, of course, we have the PermeaPad technology is based on a layer of cellulose hydrate or regenerate cellulose that has a cut of around 15 to 14 kilodalton. So much larger molecules will have problem to permeate. So I would say medium, small molecules.
Okay, another question is for the adipocyte slide, as neither solubility, top right, nor Papp, bottom left, was nicely describing the in vivo red permeability on its own, what parameter would you suggest to explain the in vivo data? For example, cumulative acceptor concentrations?
Exactly. This actually take back to the other question of before. If you have a very poorly soluble and permeable drugs, that I guess it would be the case. Yes, accounting for the total amount cumulated. Yes.
Is there a predictivity for pH-dependent permeability with PermeaPad?
Yes, we have several data on this. Some has been generated just lately from in my work here. We can even for drugs that has a very slightly effect on pH, we can account for changing in pH. Absolutely, yeah. This is actually working very well.
Okay, great. So I think that has been the questions. Before we end the presentation, I will ask you to take part at our survey we just posted the link to. That would be very much appreciated to improve the presentation and to talk about things that you are interested in the further webinars. Then, yeah, another reminder for the next webinar, which will be around in 4 to 6 weeks, about the in vitro permeability experiments, where we're going to talk about the experimental approach and the data interpretation. This seems to be a very crucial part from the feedback we have received so far from sample users and customers alike. Yeah, again, Max, thank you very much for the presentation.
My pleasure. Thanks a lot for inviting me.
Yeah, and yeah, stay safe and see you at the next webinar.
Thank you.
Bye.